Right, welcome everyone. So today we're continuing our journey on deep learning for image analysis, and we're really fortunate to have Juan Caicedo with us from just across the street at the Broad Institute. So he's a New Schmidt Fellow, and he's been working on image analysis using all kinds of machine learning, including deep learning. And truly, Juan, it's a pleasure to have you.
Thank you so much, Manolis, for having me here. It's very exciting for me to be sharing the research work that we have been doing using deep learning for image analysis in biology. I'm going to share my screen so you can see the slides. Great. Can you see my screen?
Yep, very well.
Nice.
I see your pointer too, which is perfect.
Perfect. So what I'm going to be describing today is an area of research that we call image-based phenotyping or image-based profiling, which is kind of an approach to understanding biological systems, especially when we're designing treatments or when there are opportunities for discovering new types of phenotypes or functions of cells. So what is this all about? So turns out images, biological images specifically, can be quantified for all types of phenotypes. In this slide, you can see different types of microscopy images which reveal muscle structure, or maybe you can see the biopsy of patient tissue. And we can use any type of microscopy technique as well, including image mass spectrometry. We can capture 3D images and sometimes even time lapse. So really microscopy is kind of a technology that allows us to see cells in many different ways at different levels of resolution, including temporal and spatial resolution. So we can really see a lot of information. And one of the challenges is how can we quantify that information? Because it looks beautiful in pictures, but in practice, for making decisions about treatments or for discovering new types of behaviors in cells, we need precise measurements of what the cell is doing in order to make decisions. So here is an example of the type of measurements that we can make using images. This was a research project that was run a while ago in 2012, in which researchers were looking at a potential drug candidate for leukemia. And they noticed that, or the hypothesis that they had was, if we take cells that are sick, in this case, red blood cells, which are sick with leukemia, and they are testing specific compounds to see whether the cells can recover from the disease state. There is one property in this case, which tells us when the cells are healthy again. So in the image in the left, we see sick cells, which are treated with a neutral compound. And then in the left, in the right, sorry, we see images treated with one specific compound, which was one of the candidates, in which the cells recover their normal size. So cell size was the specific feature that they're looking at here in order to determine whether compounds are doing a good job or they are useless, because sometimes in the drug discovery process, you cannot find the type of behavior or the type of effect that you would like to have. But in this case, they found this specific compound after screening several other candidates, and the way in which they measured the impact of the compounds was by looking at images and measuring the total amount of DNA, which is what we plot in this other graph in the right. After several clinical trials, and which has been, like a long term research project, probably in 2019 or 2020, this specific drug was finally approved by FDA as a potential treatment for leukemia. And the researchers behind this specific study now have a company which is dedicated to look for new treatments or new drugs, all based on images. So this is, just a concept of how far we can go with images for designing treatments. And in the previous case, we looked at one specific feature that you can use for determining cells that are sick versus healthy. But in the more general case, if we want to compare one population of cells like this one against another population of cells such as this one, there is no obvious feature that we can point at. In this case, it's not necessarily just the size, as in the previous example that I gave. There are many more features and many more characteristics of cells that are changing from one treatment to another. And for those cases, we need perhaps more information or better strategies for extracting information from those images. And that is what we call image-based profiling. So basically, this is a way of studying the morphology of cells or the state of cells using images. And imagine that you are designing a chemical treatment or a genetic treatment as well. It really depends on what type of intervention you want to make in the cellular systems. And you make the simulations in plates. There are called multi-well plates, in which you can run multiple experiments in each of those wells at the same time. And in each of the wells, you put a different compound, for instance. You let the cells grow, and the simulation happens for several hours. And then you take pictures of the cells in order to capture the state after a certain time, after letting the compound or the genetic treatment make some effect on the cells, and you capture those images. And then the next step is the computational part, in which we take those pictures and we make analysis in order to extract quantitative information to estimate which treatments are working and which treatments are not. And this type of approach has a lot of applications in biology, primarily in drug discovery, in which you can test compounds, but it also has many applications in functional genomics, in which we can knock down genes, or we can intervene, edit the genome, for instance, in cells, and we can see how they react or what happens with the phenotypes. And that can give us information about, many types of potential treatments as well. So what I'm going to be talking about today is two computational problems that are associated to this specific image-based profiling pipeline. The first one is cell segmentation, and the second one is single cell representation learning. So let's get started with cell segmentation. So the problem of cell segmentation consists on identifying where single cells are in images. And there is one, actually there are many ways in which you can achieve that, but the diversity of microscopy images that biologists capture is also pretty wide. So if you go and look at the literature, for each image type, you can find one specialized segmentation algorithm, which is kind of not good, because anytime you change your protocol, and biologists like to be very creative about how to combine certain colors or imaging techniques in order to see more and more details about the cells. But every time you change your imaging technique, you're going to have to adjust the segmentation algorithm in order to make it work in practice. And that prevents a lot of progress in labs, because they either stick to the known type of image protocol and segmentation algorithm, or if they change anything, they have to spend more time and energy into segmenting those images. So what we started investigating a while ago is what if we make segmentation algorithms for cells that work as well as phase detectors? In natural images, we have algorithms that go and look at pictures in social media or even in our own phones, and they automatically detect faces without us having to tweak any parameter or having to do any training or something in our own, right? So those models are trained in order to detect faces in a variety of conditions, including crowds or indoors, outdoors, and so on and so forth. And they work well. So why don't we have something like that in biology as well? So we asked this question and we organized the 2018 Data Science Ball around this specific challenge. What does it take in order to make one of those, computer vision technologies to work in biology as well? So we offered a prize of $100,000 to, the team that was able to segment any type of microscopy image. And here are some examples of the 5 types that we considered in this competition. There are, of course, many more, but we included 30 different experiments with 5 image types. And the competition ran for 3 months. We got participation of 3,000 teams, and all together they ran more than 65,000 experiments trying to crack the problem of how to segment single cells on any type of microscopy image. And basically, behind the scenes, what we had to do in order to organize this competition involved 3 main steps. The first step is, of course, creating the annotated dataset, because we were expecting the teams to participate with supervised machine learning models. So in doing so, we're going to need input-output examples. So we created the dataset collecting images from multiple different experiments, and then manually annotating what would be the output that the algorithm is supposed to produce in order to obtain an accurate segmentation. So we call this the ground truth. And then after collecting a set of images with approximately 32,000 single cells manually annotated, we split the dataset in training and testing. In fact, there are 3 major partitions here: the development partition or the training partition that was shared with the public, and participants are allowed to use this training test in any way they like. And then there is the second partition, which is test 1 partition that they can use to get feedback from the system. You don't have access to the ground truth annotation for the test datasets. You only see the images. And your task is to produce the segmentation for those images so the system can tell you how accurate is your segmentation. In order to evaluate whether people are doing progress in this task, we also have to define a metric of success. And in this case, the metric is intersection over union, which basically measures what's the ratio between the overlap of 2 cells. And here is an example. Imagine the purple circle is a real cell, and the algorithm is giving us this smaller object. We can compute the overlap or the intersection of both areas, and we compare that to the union of both areas. And that's a score that tells us how accurate the segmentation is. Ideally, we want the objects to be exactly the same as the ground truth. But in practice, there is some mismatch that we aim to compute with this metric. And this is the feedback that we give to participants in order to let them know whether they are doing a good job at segmenting the cells in the test set. In the final round of the competition, they only have 1 week to segment the final test set, which is test 2. And they only can submit, if I remember correctly, maybe 3 times in that time span. And they don't get any feedback, because that's going to be the data set that defines the winner of the competition, who is able to segment the final test 2 data set more accurately according to this metric. And again, the models for segmentation in this case were supposed to be supervised training models, which basically require some example images with example outputs. So we can train a model that is supposed to learn the relationships between inputs and outputs. And then hopefully during the test cases, they only have to run the model in order to generate a segmentation map of the image that we provide as input. And notice that here we don't make specific assumptions about the model that the competitors can use. They can choose any um, architecture or machine learning model that they want, or even classical segmentation if that works as well. So in practice, the top three competitors in this competition use the following architectures. So on the third place, there was a team called Deep Retina, which used the mask RCNN architecture. And this is a classic architecture, classic by today's standards. Everything changes so quickly in deep learning. But this one was proposed back in 2014 and 15. So it's a few years old already, but it's very well established. It's a solid architecture for generating segmentations. And the way it works is that it looks at one image and it decomposes the image in sub-regions. That's the R in the masked CNN procedure. This is the regions that the system believes there may be some objects of interest. So there is one region proposal network here, which takes the features from the convolutional neural network and generates some candidates. Those candidates are reviewed again by another network, which is able to tell if the object is a real object or is not in order to discard it. And then if it is a real object, what would be the exact bounding box that encloses the object, and what would be the mask that separates the object from the background. So all those components are here in the mask RCNN architecture. And with that, this team was able to reach the third place. It's kind of good to see that, reusing computer vision techniques properly, you can achieve high accuracy for cell segmentation as well. Many other teams used this same architecture, but they were not able to make it work as accurately as this specific team. So that also tells us that it's not just about the architecture, it's also about the way and the rigorosity in which you run cross-validation experiments to make sure that the algorithm is working properly.
So Juan, is this basically what made the difference? Is it just the setup, or are there any pre-processing steps that they did differently? I'm just wondering if you could give us some intuition as to when teams are more successful or not using the same underlying architectures.
Yeah, that's a great question. Unfortunately, we don't have the details of all the other competitors that were using the same algorithm. We know there were like a population of people using this algorithm, in fact, using the same code, because in the forum, they share information. You can see people talking and exchanging ideas there. And many people were using the implementation that this same team used. But we don't know their exact configuration, and that kind of limits us to understand what was the difference. We had the opportunity to interact with this person, and they shared all the configuration that they used. And according to their experience, there are a few parameters that make more of a difference for images of cells. And maybe those parameters were missed by the rest of the people. So calibrating those parameters.
I think in general, being able to demystify it and basically turn it from an art to a science and say, here are the sort of steps that you need to take. But if it's really that the steps is that it's an art and you have to try all these different parameters, then maybe
it seems to be the case. We don't have more insights into that. Everybody was doing experiments at the same time. Maybe this person had more time than the rest also. So we don't know the conditions in which the others were.
It's already very helpful. Thank you.
Sure. So in the second place, what we had was another type of architecture. And I think this is also kind of interesting because, again, it tells us that maybe the exact architecture is not as important as the way you train it. And the architecture here is an image pyramid network. Basically, in this case, we take the image as an input, and then we compute multiple feature maps that are used to generate several intermediate outputs. So you get feedback on all of the scales of the image, in order to improve the features are different resolutions. At the final layer, you are aggregating information from all those resolutions in order to generate the specific output. I think one of the innovations of this specific solution was not the architecture itself, because it's also one of the well-established architectures in computer vision. It was the way in which they modeled the output. Instead of predicting binary masks, which is kind of the practice for this type of problem, they suggested to predict some sort of a distance map or angle maps in which they kind of a gradient, if you like, in which we can measure the distance of one specific object to the center of the cell in different directions. Like what is the direction, sorry, what's the distance in the 45-degree direction from here to the center, and so on and so forth. So they created some encoding, manually engineered, I guess, of the outputs in order to get more precise measurements of where the exact object is located. And with that, they reached the second place in the competition. I think the major innovation was the way in which the outputs were encoded.
Has this been used before, or was that a novel idea that they came up with?
I think it was a novel idea, but some other works have followed up after this. And now there are a couple more papers that evaluate what's the value of using this. And apparently it's very robust, especially when you have crowded images with a lot of objects. Being able to tell what's the distance of the edge of one object versus the center seems to be more informative.
In the class, we talked a lot about representation learning and the creation of almost dummy tasks that can force the system to learn representations. This seems like a very interesting idea in that vein, basically.
Very interesting.
Yeah, where by tricking the system to sort of predict something specific that you can measure on the image, and then use almost self-supervision.
Nice.
I think you can sort of extract this kind of representation. I also like very much the hierarchical nature, and that's something that, again, has been very successful in the field. So, constraining somehow this multi-resolution approach.
Yes, this is a very nice type of architecture. Unfortunately, the person behind this work didn't follow up with a, publication with more details.
Yeah.
But others have looked at their code and have tried to formalize a little bit more those ideas. Yeah. And there are a few papers, related papers, that I can point to as well in case anybody's interested.
Great, thank you so much.
And finally, in the first place, the type of architecture that they used is a classic one. It's called the unit. Maybe you have seen it in other works. The basic idea here is that you take the image and you compute representations all the way down to a lower resolution or even a single feature vector. And then from there, you decode that representation back into a full resolution image. It's a encoder-decoder architecture with skip connections between the multiple resolutions. You can share information from the input or the encoder path all the way to the decoder. So they kind of communicate in a way, basically by copying information from one to the other. It's a very simple architecture. The innovation here is not so much the architecture, because it's also pretty old and kind of simple as well. The innovation that they made is that they replicated this exact architecture in 32 different models, basically, because you can choose how many layers or how many filters or what's the resolution of each. So they have an ensemble of 32 different units. And here are some of the names of those units based on resnets, based on densenets, even based on inception resnets and things like that. So they have a wide range of different architectures in order to form the encoder and the decoder. And they trained all of them simultaneously. So this is a really large scale type of ensemble. I think it's way too computationally involved for the type of task that we wanted to solve. They won the competition, and I think an ensemble is useful for winning competitions, but in practice, simpler models seem to do a great job. The difference in performance between this ensemble of units and the second and third place is really minor, and we think that is probably not significant. But the computational load is definitely a lot more.
Just to understand the model on the left, are the different networks talking to each other, or every single one of them is trained independently, in parallel, but independently, and then they all make a prediction in the end, you just do some ensemble approach?
That's a great question. Yes, they are independently run and independently trained. And after you do that, you take the test image and you run it through the 32 different neural networks and aggregate the output somehow in a final post-processing step.
I'm curious whether they sort of looked into the black box of each of them, actually looked into the individual methods to basically see if there were some that were constantly outperforming the others.
That's a great question. I don't think they made any specific analysis, probably because of the short amount of time that they had for the competition. I think this is also one of the limitations of competitions, that given the short amount of time, sometimes there are some details that cannot be studied in detail. And I think that's a great question. I don't think all of the neural networks are equally important. So it would be nice to prune the ensemble and only leave the couple of models that are giving more accuracy in practice.
Did they also write up their method?
No, they did not.
No, okay. But if you can send us the papers, I mean, I'll follow up with you. That'd be great.
Absolutely. One thing that all of them had to do, though, was to kind of document their approach and share the code.
Yeah.
So the code for all of those approaches is publicly available. And in theory, anybody can, take the data set, which we created, and that data set is publicly available, is public domain. So you can use it for academic or commercial applications. There is no restriction on the usage of this data set. And you can also download their code and test the different methodologies that they tested in order to do some sort of a meta-analysis maybe, or something like that. That would be great.
Very helpful. Thank you.
Cool. So with those three, one of the things that we really wanted to evaluate as well, beyond accuracy only, is what is the improvements that we get in facilitating biologist research. And in this case, imagine that we have 100 images in these 5 types, and you have a biological lab, and you have to segment the cells in those 100 images this week. So what are the options? One of the options is we can hire a data scientist that can take a basic unit model and train it with the training sets that we have, and then run one unit for each different type separately, because that was the practice at the time. Like, everybody was optimizing their algorithms for specific types of images. And this is what we replicated and what we did. And what we observe here is, first, this person is going to spend about 20 hours of work optimizing the 5 units for the 5 image types. And second, we can see how accurate you can get by running an independent model in each of those images. So what we have here is the colors of those points represent one image in each of those categories. So green, for instance, there is only one green image here, which is this one, and then purple images and so on. The accuracy here is the F1 measurement, so higher is better. And as you can see, there are images that are harder to segment by those models. In average, you get 62% accuracy here.
So this image is harder to segment because you just don't have enough training for that type of image?
Exactly. I think that's one of the issues in this specific data set. There is imbalance in the type of annotations that we get for each of the data sets, which kind of represents also the real world situation. Some images are also harder to annotate because they require experts really going into, saying this is an object and this is a separate object. So we definitely had less examples of the blue and the purple, which are the harder to segment in this
But
case.
it would be helpful to know if different types of imaging techniques are more easily parsable than others.
Yes, that's also, yeah, I think that's true. In this case, the small fluorescent is probably the easiest to parse. So this one is much harder, like the pink and the purple, they are more difficult to, segment. Um, we have more detailed analysis of what are the challenges of each of those. In this case, what I'm presenting real quick is kind of the trend, especially when you are using different approaches. Uh, Training one model per image type is one possibility. Another possibility that you can do is just segment the images using classical algorithms. And in this case, you basically have to, download an image segmentation tool and then configure the parameters. There are multiple things that you can use. And this person is going to spend less time because it's just, adjusting parameters, but is going to get pretty much the same accuracy as well. Same type of problems. Some images are harder to segment, and this is just classical algorithms without having to do any training or any optimization. If you ask an expert image analyst, they can, optimize the parameters of segmentation algorithms much more better because they have experience on what are the parameters that help me get the segmentations right. And they can get up to 74 percent accuracy, which is kind of interesting, in even less time. So having experience definitely helps. And finally, if you use the top one model in this competition, you don't need to configure anything because the model is pre-trained to take any image. And interestingly, you can also get higher accuracy. And I think this is kind of the message as well. If we combine all the, synergistic learning that you can get from all the images, you can do it more accurately. And at the end of the day, you only have to train one model that you can share and use as if it was a face detector. So there are more recent works that have specialized in specific aspects of segmentation. And I think we are now reaching the goal of having a kind of universal segmentation tool for cells. There are at least 3 methods that you can find on the web: nucleizer, cell pose, and this one is called Mesmer, which was recently published, maybe last month or so. All of them are pre-trained models, and surprisingly, all of them kind of use strategies that people used in the competition as well. Masked CNN in the case of nucleizer, UNets in the case of cell pose, and feature pyramid networks in the case of Mesmer. So we are kind of making progress as a community for segmenting cells automatically. We are not just segmenting the nucleus, we now segment also the cell body or the cytoplasm of the cell, which is, of course, a little bit more challenging. And this is what we get. There are still some open challenges in segmentation. The first one is collecting larger publicly available data sets. Ours is an example, but it's kind of small compared to, other data sets in computer vision. And also the ability to learn from, fewer examples, maybe using active learning in order to optimize the time of experts spent on identifying objects and making sure that the model is able to learn what's important in those images without having to, really go into the large scale regime. Okay, with that, I'm gonna switch gears to the second topic, which is single cell representation learning. And I think I'm not gonna have too much time to go into details, but basically going back to the idea of profiling, we want to transform images into, information that we can use for making decisions. One step is segmentation, which we just talked about. And the second step is measuring the phenotype of cells. That was the initial motivation, like what is going on with the cells? And we need to measure those properties. A classical approach to measuring morphology is to, just use area, shapes, the size of the nucleus, or very specific engineered measurements. But as we know, machine learning methods may be able to learn features that we cannot describe easily as well. And that is the case with cells. Just looking at the cells sometimes is complex enough, and discovering the features that make a difference between a population of cells is something that the machine learning methods may be able to help us better. Turns out there is one important caveat here, which is we are dealing with perturbation experiments. So what that means is that we're going to take cells and we are going to treat the cells with compounds, some compound structures, like the chemicals, and we apply those compounds to cells in plates, and they give us a morphology, which is what we observe in the images. But those compounds are usually organized in batches, like plates, and the batch makes an effect on the morphology as well. So if it's in the morning, maybe the cells are going to be happier. If it's during the night, they're going to be sad. Or if this is another person making the experiment, some microscopy perturbations. At the end of the day, this is very sensitive cell biology models. So any type of minimal perturbation in the batches is going to make an effect here. And what we want to discover really is what's the phenotype that explains the effect of the compound. We cannot measure the effect of the compound directly. We want to observe it through the morphology, but the batch effects sometimes generate noise and sometimes confound our understanding of what is important in the phenotype of cells. Oh, by the way, we don't have ground truth annotations here. This is a major difference with respect to segmentation. This is not a supervised learning problem. We don't know what's the phenotype. At the end of the day, this is drug discovery, and we don't know if the compound is going to work or not. So there is no ground truth. And hopefully we can use methods that need no supervision at all. Here we are using a weakly supervised learning method, which uses information about the compounds. So basically we take a cell and we run the cell through a neural network, and we ask, can you tell what was the compound that was applied to this cell? Because that's part of the experiment, and we know what was the compound. What we don't know is if the compound works or not. So the network has to make this classification decision, but we don't care about the classification because we already know what was the compound applied to the cells. What we want is the features that the network comes up in order to organize those cells in a meaningful way, because those features are going to tell us whether the compounds are similar or not. And how do we evaluate that? Imagine we get 13 compounds in this case. So each of the gray nodes in this graph is one compound. And we have a population of cells in each of the compounds that have been aggregated. Let's say this is the mean of the morphology according to the features in the network. So what we want to do is to be able to understand which compounds are similar to each other. So we can connect this graph, all compounds against all compounds, and see whether the connections that we observe in the graph are biologically meaningful or not. And this is only for benchmarking purposes in order to understand whether the discoveries that we would make, because this is kind of a simulation, are biologically significant or not. If the feature representation that we get is useful, we should be observing clusters of compounds that share similar biological effects. That's pretty much the goal here. In practice, when pharma companies are actually running this technology, they don't know if the connection is real or not. So they have to follow up with another experiment, maybe more complex, not just images, maybe sequencing or something else, or maybe with real patients as well. But the goal of using images is to reduce the search space to a few compounds that are useful. So when we evaluate this in different experiments, we observe that there is a large difference between the deep learning features and the classical features. In the x-axis, we have the total number of connections that we observe as a percentage in the graph. And in the y-axis, we observe how much enrichment of truly biologically meaningful connections we observe. And deep learning gives us an edge, a large margin of difference, connecting compounds especially. We have 1,600 drugs in this case, and we want to see if they are biologically connected or not, just by looking at the images. And these results are indicating that that is the case. Other types of usages that you can have based on cellular features, determining the impact of variants or mutations in cancer. Sometimes when cancer patients have a tumor, they now routinely sequence the tumor, and you can tell that there are many mutations, but we don't know if the mutation is driving the cancer or is just a passenger mutation that is an effect of the cancer, but not really generating the cancer. So in this case, we compare the original type of the gene versus the mutant just by looking at images again, and we can see that there is a difference just by eye. The question is, how can we quantify it? And we can compute features, we can compare populations of cells. In this case, the blue, red, and green populations are being compared, and we can measure what is the phenotypic similarity between those in order to estimate whether the mutant is really driving the cancer or not. So this is another application of measuring cell morphology. And I was talking about batch effects. This is, something that we have to watch during the learning process. If we don't correct for batch effects, the data is going to have one structure, which basically correspond to batches. This is the colors here in the bottom row. When we correct for batches after the fact, we can eliminate some of that structure, but it's still present there. Ideally, we should use more principal methods in order to prevent batch effects. And this is one idea that we're testing recently, which is domain adaptation. We basically can put another classifier. The label here is this is the neural network, and it has two heads. The first head classifies the compound because it's something that we can use for learning features. The second classifier determines the batch, and we don't want this information to influence the features that we use. So we use the negative information of that gradient in order to destroy any potential information that comes from the image and is associated to the batch. Using that approach, we can correct batch effects. In this case, the top row has neural network learning features without batch correction, and the column in the right is indicating the batches, whereas the column in the left is indicating, the column in the left is indicating information without batch correction. So as you can see, it's all mixed up, and we want to separate batches from phenotypes in order to reduce the confounders. Ideally, it should look like this. Batches shouldn't influence any of those groups, and the phenotype should be clearly determined by the network, which is what we get with the gradient reversal layer. Here are some open challenges on representation learning, which involve batch correction. It's still a problem, and explainable models, because the features are usually not meaningful when they are learned with deep learning. So we really need to give a push to the field and fix that as well. So with that, all I want to say is images are a great source of information, and they have a lot of things that we can do, a lot of open challenges as well. But hopefully they're going to be helpful for biological discovery, which is what we really want to push. Thank you so much for your attention. And I think that's all I have for today.
Thank you so much, Juan. It's really a wonderful, wonderful pleasure to have you. Thank you so much for your insights. And I look forward to interacting with you, and I will follow up on the various links as well.
Awesome.
Thanks.
Absolutely. Have a
Bye.
wonderful afternoon, everybody.
So Jackie, can you please make me the host again? So our